In this video, I will be discussing what meiosis is, what crossing over is, when it occurs, and why meiosis leads to so much genetic diversity in sexually reproducing organisms. Interphase is a growth period that the cell goes through before division, where it grows and readies itself for the process of meiosis. During the G1 phase of interphase, the cell goes through a growth cycle. During the S phase of interphase, the cell goes through DNA replication. The last part of interphase is the G2 phase, where the cell goes through more growth. The first phase of meiosis 1 is prophase 1. The nuclear envelope disappears, DNA coils tightly, and homologous chromosomes come together. Crossing over occurs, which is when segments of DNA of the same gene from the mother and the father are switched, resulting in genetic diversity. During metaphase 1, the homologous chromosomes line up at the metaphase plate. They can line up in any order with no factors affecting which way they come together, which is called independent assortment. Microtubules then form spindle fibers which attach to the centromere of each homologous chromosome. During anaphase 1, the spindle fibers pull each homologous pair of chromosomes to opposite poles. During telophase 1, the cell cinches apart into two separate cells, and the nuclear envelope reforms around the daughter nuclei. Between telophase 1 and prophase 2, no DNA is replicated. The same processes happen during meiosis 2, except instead of pulling apart homologous chromosomes, the spindle fibers are pulling apart sister chromatids. The end result of meiosis 2 are 4 non-identical haploid daughter cells.